Hello, thank you everyone for making the time to attend Deuterium's webinar, Empowering Small Cell Multi-Omic Data Analysis with the SciDAP Online Platform. Joining us to present will be Dr. Barsky, professor at Cincinnati Children's Hospital and co-founder of Deuterium. The webinar will begin in just a few minutes. Before the presentation starts, however, a few housekeeping items. By default, your microphones will be muted for the duration of the presentation. We are going to keep this webinar to about 45 minutes. If you have questions, please use the questions section of the control panel that should be up on your screen to enter them. If there's time at the end, we will be answering the questions. If we don't get to your question, don't worry, we will answer all questions via email within a few days' time. I see a few more people logging in here, so I'm just going to pause for a few seconds before we get started. Okay, great. Even though some people are still logging in, we will get started. Hopefully everyone will be on by the time the content starts. My name is Jillian. I'll be moderating the webinar. Welcome to Empowering Small Cell Multi-Omic Data Analysis with the SciDAP Online Platform with Dr. Barsky. And we are going to start out with a poll question for you. So you should see on the screen what NGS-based techniques do you use and be able to select an answer. We encourage you to participate, and let's get the vote results up to 85%. Okay, great. Let's show those results. Okay, Dr. Barsky, over to you.
Thank you, Jillian. Thank you for the introduction. So today we will be talking about our data analysis platform, SciDAP, and how to use it to analyze single-cell multi-omic data. We started our platform initially for ourselves. As Jillian mentioned, I am an associate professor at Cincinnati Children's, where we study epigenomics of T-cell memory. And of course, with all the omics studies, we produce a lot of NGS data. This is why we needed a quick way to analyze it. So normally when researchers do experiment, they can make libraries, and then the data, the libraries go to the sequencing core, which produces FASTQ files, which can go to bioinformaticians. Now with a lot of projects that they're working on, sometimes it takes very long time for bioinformaticians to process the data, and it can take months for researchers to figure out whether the experiment worked or not. In order to solve this, we wanted to automate this initial data analysis, giving the opportunity to researchers to not only figure out whether the experiment worked or not, but also browse the data, make initial conclusions, and maybe even get some images for publications. So SciDAP can process these FASTQ files into results, but we do not, of course, exclude bioinformaticians who can continue working on this data and do additional analysis that might not be automated. So the advantages for biologists, of course, is faster turnaround time. They don't need to learn how to code, how to type on the black screen in command line. The pipelines that we're using are state-of-the-art. They are what everybody else is using. We typically do not invent new tools. We just make existing tools easy to use and combine them into pipelines. We produce quality control data, which help researchers troubleshoot experiments. Researchers can explore data and change parameters of analysis, for example, what fold change you are interested in, or as you will see, what kind of integration technique for single cell samples you want to use. And our pipelines can produce interactive publication quality images. The advantage for bioinformaticians is that the pipelines we use are all written in so-called common workflow language. They are transparent, reproducible, and portable. In fact, almost all our pipelines are available on GitHub, and they can be run outside SciDAP, but of course, there will not be our nice user interface. Another advantage is that all data and metadata are stored together, and this allows everybody to access the same data, and bioinformaticians and biologists will be working from the same initial preprocessed data set. And nobody needs to run around with hard drives, look for data on somebody else's laptop, and so on. And as a PI, I really appreciate these advantages, which is immediate access to data. I can see whether experiment worked or not and how the analysis was done. And more importantly, when lab members leave the lab or the company, both the data and the methods that were used to analyze them stay in the lab so that additional data can be easily analyzed the same way. Here are a couple of screenshots. This is how you start analysis, add a new sample. You select what type of sample, in this case ATAC or scHMultiome. You provide a description of your experiment, select the genome, and attach FASTQ files. And by attach, I mean it can be either drag and drop, or FTP, or even URL or SRA number from NCBI. Once the sample is processed, you will get a list of your samples with some, in this case, mapping information as a pie chart. If you click on the samples, you will get detailed QC information, such as mapping and so on. There are some QC plots, and you can browse the data on the genome browser. And of course, you can compare different samples on the genome browser. In this case, I'm showing 3 scHMultiome experiments. Overall, SciDAP has some 90 pipelines, and they can be used to process data obtained by various methods, including single cell, like single cell RNA-Seq, ATAC-Seq, and Multiome. We're adding single cell VDJ. For bulk transcriptomics, we have different flavors of RNA-Seq, paired, single, stranded, unstranded, quant-seq, and microRNA-Seq. We, of course, have epigenomics methods like bulk ATAC, scHMultiome, cut and round, cut and tag, DNA methylation, genomics variant calling, and for microbiome, we have CHIME and Kraken for taxonomic classification of the organisms. Very briefly about our pipelines. So they, as I mentioned, they are written in common workflow language, and this is a way to make pipelines very reproducible and well described. So for each step in the pipeline, they're called command line tools. We have description of what is the command, what is the version of the tool, what are the inputs and outputs. And because we have these detailed descriptions, it becomes easier to combine these tools into pipelines, connecting inputs and outputs. And in fact, there are even graphical editors for CWL. Deuterium also enriched CWL with what is called CWL extensions. And this is what tell SciDAP how to create input form from inputs and how to present the results. For example, if the pipeline produces scHMultiome genomic coverage, then the platform would know that this needs to be shown on IGV genome browser automatically. So this makes it easier for us to add additional pipelines to SciDAP. I think we missed
Okay.
Paul question. I think we wanted to ask what Can you go with the next one?
Sure. So our next question is, what kind of hardware setup is ideal for your laboratory? We encourage you to vote in our poll. Okay, we still have votes coming in. All right, thank you. And here are our results.
Thank you for giving us this information. So with SciDAP, we can accommodate different types of hardware. So when you use SciDAP, you use a browser on your laptop to go to scidap.com, and scidap.com would provide a user interface that you will see in your browser window. However, the data processing happens on what we call satellite server, and this server can actually be controlled by us, controlled by your organization. It can be a cluster, it can be cloud, and this is where the data processing happens. So raw data are loaded directly into satellite server, they're processed and stored there, and when you want to look at the results of your analysis, they will be loaded directly into your user interface from the satellite server. So if you want to keep your data private, we allow this, and in this case, the data would never leave your organization. However, the user interface and the pipelines are provided by us, and we can give you updates of this every time. And pipelines can come from GitHub, which can be either our GitHub, in this case, the pipelines will be available to everybody, or if you have your own pipelines that you don't want to share with others, then it's possible to keep them private. And the next poll question is, we actually wanted to know how important it is for you to keep the data within your organization.
Here, next poll's up. Okay. Sharing those results, it looks like we're at 100% one. Yes, that is critical.
Thank you. Today we will talk about analysis of single-cell data, but as I mentioned, SciDAP can be used to analyze bulk data, and you can find information about how to do it, of course, on our website. But also we have YouTube channel where we record videos and how to step-by-step guides how to do the analysis. Also, we have some discussion on our Medium blog, and you can easily Google for YouTube SciDAP or Medium SciDAP to find them. Now we will move to the topic of today's webinar, is how to do single-cell data analysis in SciDAP. And this I will probably not be able to cover all the details here, but we have recently put a preprint on the bioRxiv server where Michael Kotlar, bioinformatician at Cincinnati Children's, describes using single-cell data analysis in SciDAP in great detail, so you can refer to this as a guide when doing the analysis. So why, first of all, let's talk about why use Multiome. So before 10x developed Multiome, you could do single-cell RNA-Seq and single-cell ATAC-Seq experiments separately. And what happened there is that you could get your transcriptome data for your cell types that are in the samples. You could get accessibility, and then you had to infer what cluster in the transcriptome analysis refers to what cluster in the chromatin accessibility analysis. And this inference has always been problematic because this assumed that genes that are associated with open chromatin are expressed. But as we know very well, it is not true at all, and there are a lot of poised genes which have open chromatin but are not expressed. And so this connection was always very tedious. Now with the multiome, we don't need to do this guessing. We can get actual information from the same cells, and this allows us to infer true modes of regulation for these genes. Another important difference is that in single-cell RNA-Seq, you are looking at genes mRNA that are present in the cells, and this includes both the nucleus and the cytoplasm. Whereas in multiome, you are looking just at the nuclei. And this makes responses to regulation much faster because there is much less of influence of RNA stability on the level of RNA there. How these experiments are done? So we prepare our nuclei. Transposition reaction is done before loading on 10x chromium controller. And we have this suspension of nuclei that is enveloped together with 10x barcoded beads in the oil drops, and creating this single nuclei germs, as they call them. The 10x does RT reaction, reverse transcription reaction in the gems, and then breaks up the suspension and preamplifies and eventually amplifies, creating two libraries. One library for gene expression and another library for ATAC. And these libraries are sequenced independently on usually on Illumina, but could be other sequencers. And when you sequence, you usually use both indices. And so of the sequencer, you can get 4 FASTQ files. So for gene expression, you don't need all 4 files. You need only read 1 and read 2. So read 1 contains cell barcode and UMI. So this is to avoid the duplicated reads. And read 2, which contains insert, which is the mRNA sequence itself. Indices are not needed. They're only used to identify the samples within the flow cell. In case of ATAC, we need 3 of the 4 files. And in this case, read 1 and read 2 have the transposase cut sites, whereas I5 index contains 10x barcode, and you can see the information on the length of the reads. I7 is not needed and only used to identify samples. These are 10x recommendations for how deep to sequence per cell, and usually your core facility will tell you how many cells they encapsulated. So once you have the FASTQ files, you can start the analysis. And in SciDAP, the data are stored in so-called projects. You can think about it as folders which contains information about both the samples, the analysis, and the pipelines that are used to analyze the data. And the purpose here is to make sure that samples that you are comparing are analyzed by the same set of pipelines so that you are not comparing apples to oranges. So to start new project, you use this new project button. You will get to the project properties window where you can provide a description of the project and select a set of pipelines. You will have several collections of pipelines, in this case 5, but initially you will have most of the users only use our global repository, but it's possible also to connect your own GitHub and use your own pipelines. So this is overview of the pipelines for single cell analysis that we provide. So SciDAP allows to do both single cell RNA-Seq, single cell ATAC, and single cell multiome data analysis. And some of the pipelines are used in more than one process. And but here you can see how you can move through the pipelines from FASTQ data to clustering, as you can see here. We have also the list of pipelines in our preprint, and I invite you to refer to it when adding the pipelines to the project. So once a project is created, you can add samples or analysis by clicking this Add Analysis or Add Sample button to the project. And the difference is that a sample is actually something that has been in the tube, whereas analysis is something where you do comparison or processing of the data after the initial processing is complete. So this is the view, what you will see after you click Add Sample. So you select the pipeline, and for single cell multiome, it will be Cell Ranger ARC gene expression plus ATAC. You select the sample name, you select the genome, and you attach the FASTQ files. Remember, we need FASTQ 5 of them. And when you click Attach File, you will get this window. So you can either drag and drop your FASTQ file into the current sample folder. You can use FTP link to the FASTQ, or you can attach the sample from URL. In some cases, it's also possible to use SRA identifiers, SRR numbers from NCBI, but unfortunately, NCBI did not come up with a uniform way to attach, to store the single cell data, so sometimes it doesn't work. And for this, we have a special pipeline that can download data from NCBI. It's called FASTQ download, and it's also described in the preprint. So once you add your samples, you will have your list of samples, and I will be using some examples from my academic lab, where we looked at control and knockout activated fed resting cells, and some from this preprint paper where we looked at published data about resident memory cells. So once the data are processed, you can look at the results, and here is a short video describing them. Oh, sorry, I think I need to go one back. So yeah, once you can click on the sample, and this overview plot will show you basic statistics, which is how many reads, what genome is covered, how many peaks, and so on. Also, there are you can look at QC reports, including FastQC and the reports produced by the Cell Ranger pipeline. So if you click on cell range summary, you will get the cell range report. It shows you the number of cells in your sample. It shows the clusters, depth of sequencing for clusters. You can look at joint or separate and decide whether this experiment worked or not. So for ATAC, you get read fragment sizes, enrichment around TSS, and for RNA-Seq, you get coverage as well as the number of reads. In addition to cell ranger report, you can also use interactive cell browser, where you can look at the clusters identified by the cell ranger pipeline. You can highlight the clusters, you can highlight the genes. If you go to this gene tab, you can look at gene expression of this particular sample. So since we have usually several samples in experiment, you may need to aggregate the data in order to analyze them together. And this is done with cell ranger aggregate pipeline. So in this pipeline, you select the samples that you want to aggregate here using a Dropbox. So you provide the name, you select the samples you want to aggregate with these check marks. In the advanced, we have a parameter for normalization. And once you save sample, it will do the processing. So for normalization, there are this is kind of an important step, and cell range aggregate provides two options. One is to use depth normalization, and this will randomly remove reads to adjust sequencing depth for all samples. And this is recommended when using the next loop browser to view the data right after the aggregation, but will result in data loss. If you do no normalization, then the data will not be normalized or integrated, but this can be done in the next steps with Sura. Aggregation pipeline will again produce aggregation summary report from 10x. It will show you total number of cells as well as per library information, and you can review it here to understand how the normalization and comparison work. And of course, you can look at the cell browser on the interactive results. So after this, we start analysis with Sura, and we're starting with the cells that have not been filtered. Next, we filter cells on both RNA and ATAC quality parameters. Then we do dimensionality reduction separately for RNA and ATAC, and this can be done in any order. Then we do clustering for, you can do based on RNA, based on ATAC, or you can combine modalities and cluster on both RNA and ATAC using weighted nearest neighbor clustering. So let's start with filtering step, which is pretty important. So this is one example. So before filtering, we had some 14,000 cells coming from 4 samples, and dots here are cells. They are colored based on the samples. And on the y-axis, we have a number of UMI from ATAC, and on the x-axis, number of UMI from RNA. So before filtering, we had these 14,000 cells. We start by removing cells that have too much transcript from mitochondrial cells, and these are likely bubbles that came straight from basically mitochondria without the nucleus floating around. Next, we want to get the cells that have at least 500 UMI reads per cell. Next, we want to remove the cells that have data for less than 250 genes in the gene expression modality, but we also want to remove the ones that have too much because they are likely to be doublets. And finally, we filter on ATAC-UMI, and we filter and remove more, the cells that have fewer than 3,000 ATAC-UMIs. And so we ended up with about half of the initial cells. So these are key parameters for filtering. So first is we can redo peak calling using max instead of what SciDAP do automatically. We can remove doublets using SC doublet finder, and you can select whether to remove doublets based on RNA, on ATAC, or both. These are the parameters that we have discussed. So this is minimum number of RNA reads, minimum number of genes, maximum number of genes per cells, how to identify mitochondrial genes in this particular genome, maximum mitochondrial percentage, and minimum number of ATAC fragments in peaks per cell. So these are if you go into the filtering sample, this is how it will look. So these are QC measures before filtering, and you can see genes per cells, read per cell for each of the 4 samples that we have. And then you can look at these same values for after filtering, and you can look at what is the number of cells that are remaining and how they are interacting with each other. So this is one of the summary views. So this is a Wiener plot showing the distribution of RNA or UMIs, genes, and so on per sample. This is again pre-filtering. So now here we're looking at number of UMI per cells and gene per cells. So we usually want to kind of select one main peak here, but sometimes you can have cells with different RNA content. And for example, you can have resting T cells which have relatively little RNA, and you can have, I don't know, macrophages which have relatively more RNA, and so you would have bimodal distribution, and depending on your scientific system, you can set these parameters. We also have an option to automatically set those, but it works well only if there is a unimodal distribution like shown here. This is a parameter that is important for ATAC. So here we filter cells that don't have enrichment of the reads around the transcription start site. So transcription start sites are more likely to be open, and so they will be digested by transposase. And so the cells that got digested will have this enrichment. The cells that did not get enrichment, that did not get digested, will not have enrichment, and so we are trying to remove the cells, and this is comparison of high or low enrichment cells, and low enrichment cells are removed. So we also use this composite graph to look at the filtering. So this color, this individual cells, the color shows percentage of mitochondrial reads below or above 20% in this case, and we distributing cells on TSS versus UMI, and you can see we're taking cells in the top right quadrant. This is a similar plot comparing RNA reads and ATAC reads per cell, and again, we're taking top right quadrant here. Of course, many of these quality control measures are correlated with each other, and for this reason, we provide this PCA plot, which compares these measures. For example, filtering on the number of genes or on the number of UMIs will probably remove mostly the same cells. But for example, filtering on mitochondrial percentage will remove a different collection of artifactual cells. Next steps we call the pipeline dimensionality reduction, but what it is supposed to do is to normalize and integrate the data so that the same cells will fall in the same cluster, even though they come from different samples and might have been sequenced to different depth. So in here, I'm showing you three UMAP plots. This is from a published paper which looked at T cells taken from 4 donors, and the color in A and B are the donor, and you can see here that if we just aggregate the cells without normalizing and integrating, then the cells will cluster separately, and in fact we often see this problem even in published papers where the data have not been properly integrated, leading to incorrect conclusions. If you do integration properly, then the cells from different donors will fall in the same clusters, and you can see this is a good quality integration. And then you can make a proper conclusion. So, for example, the orange here are resident memory cells, and the blue are circulating cells, and you can properly separate them if you're looking at and combine different donors together. So these are some of the key parameters that go into dimensionality reduction pipeline. So first is integration method, and this can be done using Seurat SC transform or single cell harmony. Then we can decide how we want, if we're using harmony, how we want to correct for batch effects based on data set, based on time, based on something else. You can also decide whether you want to remove the influence of cell cycle, and we have 3 options. Do not remove. In this case, it's possible that cells will be clustered based on the cell cycle stage. You can partially remove. In this case, difference between cyclic and non-cyclic cells will be preserved, but cell cycle stage itself will not be. And you can also regress mitochondrial percentage or provide additional metadata. This pipeline can also be used to filter the cells. So, for example, you want to reanalyze an additional. So, for example, you do initial clustering of, let's say, PBMC, and then you want to focus on the T cells. So you can select cells belonging to the T cell cluster in the UCSC browser, select the barcodes, and subcluster them by providing those barcodes. So next step is would be clustering. So in the dimensionality reduction, we basically determine the shape of this UMAP plot that you will be getting. You can look how well the data are integrated. So the data from different samples should be populating single clusters. And in clustering, you can actually identify the clusters within this UMAP. So these are clustering results, and I have a little video again. So we can look at our clusters. There is this silhouette plot which shows how likely is that the clustering is correct. You can look at distribution of cells by sample or by group. You can also look at genome coverage for and gene expression for the samples for the genes you are interested in. So, and you can provide the gene names, and you get the accessibility and gene expression per cluster. And finally, you can go into interactive UCC cell browser, and you can color cells by identity, by condition, by technical parameters like PMI, peaks, enrichment score, and so on. Or you can also go to this feature panel and color the cells by the gene expression. You will get a number of static and interactive plots. One that people like to see is markers for clusters, and you get them in the form of heat map. So the column are each cells, and the clusters are marked here, and this also marks condition and identity of the samples, and these are the top marker genes that identify these clusters. You can look at how cells fall between clusters. So so per group, so this is so these are gene expression plots. This highlight the cells that that express certain genes that you were interested in, and also you can see gene markers for each of the clusters. In the cell browser, you can do various views. Okay, sorry. So, for example, you can click on the cluster and look at the markers that it produces. You can stain for the genes. You can look at different UMAPs and look at the same cluster in RNA UMAP versus ATAC UMAP and so on. You can get these heat maps for the markers of this particular cluster and so on. There are many very cool possibilities. You can use this split screen view to look at the clusters and highlight gene expression on the same screen. So this is FOXP3 identifying Treg cells in this case. Okay, yeah, so as I mentioned, we can produce analysis for RNA, for ATAC, and if we combine the information contained in both modalities, we can get weighted nearest neighbor UMAP, which would take information in both of them. So this will provide a lot of similar views as in the other clustering. You can look at distribution of cells per group, per samples, at both gene expression and genome coverage. These are gene markers. Again, in the UCSC cell browser, you can look at the cells identified in both. So this is highlighting technical measures in different clusters. Again, markers. Heat maps. In the split, again, in the split screen, you can highlight different things. For example, you can show how the clusters that are identified in WNN look in ATAC and so on. So on the left, we have, I think, WNN, and on the right, we have ATAC clustering. And you can see that cells belonging to one cluster in WNN can be somewhat more spread in the ATAC clustering. Yeah, so these are some of the plots. So you can split by group of the genes. This is, you can see, for example, that control cells have cells in the cluster 3 and 8, but knockout cells have much fewer of them. You can look at the same cluster in different modalities. This highlights the markers for cluster 4. This graph that you saw in the video shows the accessibility of this ATAC-Seq gene in different clusters, as well as gene expression for this gene. You can see that even though the promoter of this gene is open in all clusters, expression is only present in clusters 3, 4, and 8, which is also present as accessibility within the gene body. This is another marker present in clusters 4 and 8, or FOXP3. So next, you may want to do differential expression analysis. For example, we have resting and activated cells here, and you may want to see how expression in the cluster 0 is different between resting and activated cells. And for this, we have a differential expression pipeline where you can select the cluster that you want to look at and what conditions you want to compare. In the results, you will see the volcano plots as well as clusters that are being compared. You can provide the genes of interest for which you will see these violin plots. You will, of course, get a list of differentially expressed genes together with all changes and p-values. And these are visualized in various plots like this heat map, which you can edit colors, you can highlight annotations, fold changes, and so on, as well as clustering. So, for example, this is heat map that you can use. A MA plot highlights differentially expressed genes. You can see the ones with significant p-values. Volcano plot, I'm sure you're familiar with it. It shows p-value versus fold change. So you can highlight your genes of interest. And this is a PCA plot. It shows how the samples are distributed in the PCA, and you can color it in various ways. Finally, we have pipelines for differential abundance, and this would show the areas of the graph where you have more cells from one condition versus another condition. Okay, this probably concludes our presentation, and I will be happy to answer the question. And if you want to look at the additional information or more detailed information, please refer to our recent bioRxiv preprint. Thank you, and please contact us if you have more questions.
Great, thank you, Dr. Varsky. That was a lot of great information. As mentioned, a recording of this webinar will be sent to everyone, and we do have a few questions before we conclude. Our first question is, can we trial these single-cell workflows ourselves?
Of course, yeah. Deuterium provides a free trial. You can use Deuterium, you can use SciDAP for free for a month. And to do this, you can just go to scidap.com, register, and you can try out all these pipelines before you decide to subscribe.
Great. Our next question is Harmony or Seurat, which is the best for sample integration?
So this depends on your sample. We have both Harmony and Seurat as possibilities for integrating the data. And I guess the rule of thumb is that if your samples are kind of similar, for example, you have PBMC samples from different donors, then you want to use Seurat SC-transform. And this is because SC-transform tries to find the cells that are the same in all samples. They call them, I think, anchor cells. And they make sure that the cells are falling in the same UMAP coordinates in the same cluster. In the case of Harmony, you want to use it when the cells are more likely to be different. For example, when you are looking at differentiation curve and differentiation time curves, and you can integrate the differentiation data better with Harmony because the cells in different samples are likely to be different, and it doesn't make sense to try to find these anchors.
Okay, one more question. How long does it take to run the analysis for a 10 sample experiment, for example?
So it largely depends on the hardware that you are using. If you are using our cloud, for example, it would likely take a couple hours to process initial samples, then a couple hours for integration, and then probably a couple hours for each of these pipelines. And so since you are running them in turn, it would probably likely depend on how soon you start the next step after finishing the previous one.
Okay, we do have a few more questions, but unfortunately we are running out of time. We will answer all questions via email in the coming days. Don't forget to be on the lookout for a link to this webinar in an email tomorrow. And again, don't forget to visit our website, deuterium.com. Of course, I invite you to follow us on our LinkedIn page as well. Thank you, Dr. Varsky, and thank you again, everyone.